Hey guys, welcome back to topic 1, DNA and proteins. We're here with subtopic 1.2, which is genes and protein synthesis. Now, you've probably heard of genes before, but you probably don't know how to define them, which is really important for topic 1. So a gene is basically just defined as a segment of DNA that codes for a functional protein or an RNA molecule. Now, we haven't covered proteins yet, and we haven't covered RNA, so that's something just to keep in the back of your mind. DNA in eukaryotes consists of many genes, which contain both non-coding and coding regions of DNA. So the non-coding regions are called introns, and the coding regions are called exons. Both exons and introns are made up of nucleotide sequences. So remember, we went over these nucleotide bases. We often represent them as sequences, which is basically what base comes after the next in nucleotides. So an example here is ATCCGCCA. That's just a simple nucleotide sequence. So proteins are biological molecules that undertake specific functions within many organisms. They're essential to cell structure and to cell function. So proteins are made or synthesized in a multi-stage process known as protein synthesis. So I mentioned that it's multi-stage. There are 3 main stages that you need to know in stage 2 biology: transcription, RNA splicing, and translation. Now, the location of each of these 3 stages is shown on screen. I'm not going to go over that. You just need to memorize it. Transcription is the process whereby a premature messenger RNA, otherwise known as pre-mRNA, is created. And there are 4 steps that are very, very similar to DNA replication. And we'll cover that in a little bit. So firstly, an enzyme, RNA polymerase, breaks the hydrogen bonding between polynucleotide strands, exposing the inward-facing bases, just as what happens in DNA replication. Free nucleotides, this time RNA ones, anneal to one of the separated strands, creating a new strand of genetic material. Then, the same enzyme as before, RNA polymerase, creates a chemical bond between adjacent phosphate and sugar molecules. Now, this is known as the sugar-phosphate backbone or phosphate-sugar backbone, whatever you'd like to call it. This newly formed mRNA strand, which is a pre-mRNA strand, then detaches from the molecule, and that's the end of transcription. Now, transcription does have 4 key differences with DNA replication, and that's with the enzyme that's used to separate the strands. So it's helicase in DNA replication, and transcription uses RNA polymerase. Also, the enzyme used to create the sugar-phosphate backbone. So it's DNA polymerase versus RNA polymerase. There's the nucleotides that are added. So it's DNA nucleotides in replication and RNA nucleotides in transcription. And then the final product, which is 2 DNA molecules in replication or 1 pre-mRNA molecule in transcription. Now, we've gone on a lot about RNA, but we haven't actually covered that before, so I thought I'd just put in a slide here about what RNA is. So much like DNA, RNA is a nucleic acid, and it's made up of that same nucleotide monomer kind of structure. But there are 4 differences between RNA nucleotides and DNA nucleotides. And then, I guess, the genetic material that encompasses as well. So DNA molecules are double-stranded, while RNA molecules are single-stranded. DNA molecules, as we know, contain the bases adenine, thymine, cytosine, and guanine, whereas RNA molecules contain the bases adenine, uracil instead of thymine, and then cytosine and guanine as usual. RNA nucleotides contain the ribose sugar, which is different to the deoxyribose sugar that's in DNA, and that accounts for the difference in name. DNA is deoxyribonucleic acid, RNA is ribonucleic acid. And then the last point, which you'll rarely ever get asked, is that DNA is typically a little bit longer than RNA, but that's not really something that's covered quite a lot in stage 2. So there are 3 types of RNA that we cover in topic 1. Messenger RNA, or mRNA, is produced in translation and codes for the production of a polypeptide. Transfer RNA, or tRNA, has a complementary anticodon to the mRNA, so we'll cover what that means, and brings a specific amino acid to the ribosome. Ribosomal RNA, or rRNA, is a component of the ribosome, which is the site of protein synthesis. Now remember, all of these molecules have those same structures and same features that we mentioned before, like being single-stranded, having the bases adenine, uracil, cytosine, and guanine, and also containing the ribose sugar as well. So RNA splicing is the second stage of protein synthesis, and it's something that a lot of students always skip over, so it's really important to get your head around this. We mentioned before that there are non-coding and also coding regions of DNA, but RNA splicing is the process where the pre-mRNA has what's the products of these introns and exons separated, and the introns are completely removed from the molecule because they don't code for anything, and you can kind of think of them as junk DNA. So this process is carried out by the enzyme spliceosome, and it occurs in the nucleus. So introns are discarded as waste products, while exons are conjoined together to create a mature mRNA molecule. This mRNA then exits the nucleus through holes in the nuclear membrane, which are known as nuclear pores, and travel to the ribosome, which is the site of translation. Now, you don't need to know the mechanism for RNA splicing, you just need to know what it does and which molecule facilitates it, which is the spliceosome. So what is the ribosome? The ribosome is an essential cellular organelle, and it's the site of translation, which is a step of protein synthesis. So it's composed of protein, 40%, and ribosomal RNA, 60%. Now you don't need to know the proportions, but you do need to know the ribosome is made of RNA and protein. So ribosomal RNA is an RNA molecule, much like mRNA, and we did discuss this briefly before. And in terms of the structure of the ribosome, all you really need to know is that it consists of two major subunits, being the small ribosomal subunit, which reads mRNA codons, and the large ribosomal subunit, which catalyzes the formation of peptide bonds between amino acids. So translation is a process whereby an mRNA is used to synthesize a polypeptide, which is multiple amino acids joined together. So it's a 6-step process and it's very complicated. So I'd recommend reviewing this slide again and again so you get the hang of it. By the time you sit your first test, you should be able to recite all 6 of these steps in order if they ever ask describe translation or describe the last stage of protein synthesis or something like that. So firstly, the mRNA, which has already been spliced, lodges between the two subunits of the ribosome so that the first set of 3 bases, which is known as a codon, is covered by the ribosome. The small subunit reads the first codon, and a specific tRNA anticodon is brought with an amino acid to the ribosome. The mRNA then shifts 3 bases across so that the second codon, or second 3 sets of bases, is covered. Step 2 is then repeated again, so a second amino acid is brought to the ribosome. Then, the large ribosomal subunit, it comes in and it synthesizes a peptide bond, or an amide bond, which is basically just the name for a bond between 2 amino acids. It synthesizes that at the large ribosomal subunit. So this process then repeats until a stop codon, which is a specific set of 3 bases that stops the process, until that's reached. And then a chain of amino acids, which is your polypeptide, will detach and move to the rough endoplasmic reticulum for modification and folding. Now we'll cover more about the structure of the rough ER a little bit later in topic 2, so it's not relevant for your first test. Now I've mentioned amino acids being brought to the ribosome by tRNA and being this component of polypeptides, so I thought I'd cover what they are. Amino acids are organic molecules, which are carbon containing, and they're brought to the ribosome during translation. There are 20 naturally occurring amino acids, and each amino acid is coded by a specific codon or codons. Now, you do not need to know any names of amino acids for stage 2 bio. I guarantee you that. You will always be given them, and you'll always be given a table. The genetic code is degenerate, meaning multiple codons can code for the same amino acid. And this kind of makes sense because if we look at how many amino acids there are, there's 20, and how many possible different combinations of codons there are, because remember, 3 bases. If we have 4 bases total and 3 can fit in a codon, that's 4 to the power of 3, which is 64. So there's 64 combinations and only 20 amino acids. So this concept of degeneracy really makes sense if you think about it mathematically. So, for example, the mRNA codons GCU, GCC, GCA, and GCG all code for the amino acid alanine. And again, you don't need to know this, it's just an example. Now, I've mentioned again, polypeptides are just a chain of amino acids linked together by peptide bonds, and these are the basis of proteins. So we describe proteins using 4 kind of levels of explanation, and there are these 4 structures that are really important, and you need to know the definition of all of these. So primary structure is the sequence of amino acids within a polypeptide chain, and that could simply be like alanine, tyranine, and stuff like that. You literally just list them in order, and that's a primary structure. It's also the only structure that you would need to state explicitly for a particular molecule, because it's a little bit difficult to state secondary and tertiary structures. Secondary structure is the hydrogen bonding of the peptide backbone that causes amino acids to fold into a repeating pattern, such as an alpha helix or a beta pleated sheet. Now, these are 2 examples of potential structures, but there are many more that could be caused by secondary structure. So tertiary structure is the unique 3-dimensional folding pattern of a protein due to its side chain interactions. So side chains, or R groups, are basically the rest of the amino acid that's not involved in the peptide bonding. And it can really take any conformation within the 20 amino acids. So there's a lot of different tertiary structures that proteins can have, and because of this, tertiary structure is often used to dictate the biological function of a protein. Now, we get to that in a lot of detail in the next subtopic, so I'll leave that for now. Finally, the quaternary structure describes the interactions between multiple polypeptide chains within a single protein. And that's basically proteins consisting of 2 or more polypeptide chains. And that's all you need to know for quaternary structure.